Hereby, I open this academic ceremony in which Robin Kolpaert will defend the academic thesis of Mice and Men in ACM, Novel Models and Biomarkers for Arrhythmogenic Cardiomyopathy. May I invite you, dear candidate, to present a summary of your study and the conclusions of your thesis.
Thank you. Dear Prorector, dear members of the Corona, dear friends and family, thank you all for being here today where I present to you my thesis titled Of Mice and Men in ACM, Novel Models and Biomarkers for Arrhythmogenic Cardiomyopathy. Now, what is arrhythmogenic cardiomyopathy, or ACM? Well, if we look at a normal human heart, we see that it's beating at a normal, steady pace. But now if we look at the heart of an ACM patient, we see that it's beating much too fast and with an irregular rhythm, and that there seems to be some sort of damage in the muscle wall. And then if we look a bit closer, we see that on the right, there is still an area of healthy muscle cells, which is red, and these can still contract. But on the left, you see that these have apparently died off, and they've been replaced with this fibrofatty tissue, which you can see in blue and white. And this is pretty bad, because this is like a scar on the heart that cannot contract anymore, and it's not functional. And so clearly, this is not a good sign for these people. Now, why would this happen? Well, if we look a bit closer at the heart muscle, we see that all of these heart cells are tightly connected by these thin blue strips that are called intercalated discs. And you can imagine, as a heart needs to constantly contract and do a lot of work, these cells need to be tightly connected in order not to damage themselves. And when we look a bit closer at these intercalated discs, we see that they are comprised of all kinds of proteins that need to reach across the membrane to make these tight connections, and other proteins that reach inside the cell to make other connections, as well as little gap junctions to shuttle things from one cell to another. And it seems that in ACM patients, that there's a problem almost always within these intercalated discs somehow. And you can imagine if there's a problem with those connections, that this could damage the heart. So that's why we call it a disease of intercalated discs. But now, why would there be damage to these discs? Well, if you recall, we know that in every cell of our body, we have a copy of our DNA, which is like a packet of information. And these can create little snippets of information called messenger RNAs. And these messenger RNAs can then be translated into proteins that go into your body and do all kinds of things, such as coming together at that intercalated disc in order to perform functions. Now, it appears that in some humans that there's a sort of change in this code, a mutation, and then they can create slightly altered messenger RNAs. And the proteins they create can also be a little bit different. And as you can see in this example, because one of them is a little bit shorter, it cannot reach across that membrane anymore. It can no longer make those tight connections. So you can imagine if you can't make those connections, if they're not as strong, that they could be damaged as the heart is pumping. So this is why we call ACM a genetically inherited disease, because we know that the problem is somewhere within the genetic code. And now, it becomes even more complicated because there's not just one gene that can change and give this disease. There's a whole list of genes that can all change, sometimes even in multiple different ways, that can give the same disease. And most of them are related to that intercalated disc, but some of them not even directly. Now, we know what the disease is, but now how do we cure it? And here's the problem. There are no real cures for this disease. The best we can do is to try and focus on preventing this sudden cardiac death from people dropping and dying from it, with things like lifestyle changes, certain drugs, or implantable defibrillators. And if worst comes to worst, we can do a heart transplantation. But we don't really understand how ACM works on a functional level, so we can't really tackle it at the root. We can only fight the symptoms. And so in order to address this, we created certain mouse and cell models to try and study the disease. Okay. But what does that mean, a mouse model for a human disease? Well, imagine you have a patient with one of these mutations that causes the disease. We now have a normal, healthy mouse. So now the question is, if we add that same mutation from the human patient into the mouse, will its heart also get sick? Because if we can confirm that it also gets sick, we can study these mouse hearts to learn more about the human disease. And so that's exactly what we did. So first, we created a model in a certain gene called desmoglein2, or DSG2, which is one of those that need to reach across the membrane. And it has the same mutation that we see in a human patient. Now, what do we see in these mice? Well, in a normal, healthy mouse, they have 0 copies of the mutant, so they are, of course, looking good. And then we have a mouse with 1 copy of this mutant, and unlike in the human, it seems that this mouse is still pretty healthy. So only one copy of the mutant doesn't really seem to cause the disease in the mouse. So we thought, what would happen if we give this mouse 2 copies of the mutant version? Does that show anything? In a word, yes. Because as you can see, there's a massively engorged left atrium already at 3 months old. And then when we look at a bit older, at 6 months old, and we look at the right ventricle, we see on the top that there's clearly like these areas where the muscle cells have appeared to be have died off, they've disappeared. And in the bottom, you can see that there's like these areas with red coloring, fibrosis, which is like this scar. So this heart is starting to resemble what we see in a human ACM patient. Okay, but so we don't only want to see what's happening on the outside of this heart, we also want to look a bit closer to what's happening inside of this heart, at what their gene expression is doing. So if we look at a normal, healthy mouse, we see that they have those messenger RNAs I mentioned, and they make different kinds in different amounts. That's fine. And then if we look at the mouse with 1 copy of the mutant gene, we see that they are still pretty similar. Like for the most part, they have the same messenger RNAs in the same amount. But now, if we look at the mouse with 2 mutant copies, we see that they are much more different. So many more of these messenger RNAs are changing. And so again, we see that having 2 copies of this mutant has a much stronger effect than only having 1 of these copies. Okay, now, we know that in human patients, when you do exercise, which is normally great for your heart, but in these humans it can do much more damage. It can make the disease much worse. All right, so we figured what happens if we train these mice? If we make them run a bit, will they also get sicker? And so that's exactly what we did. We really put them on a little treadmill, made them run every day, and then after a few weeks we see how well is their heart functioning after this training. Okay, so first we will look at a mouse, regular mouse, with its heart full of blood. And we see that every time it pumps, it can still push out most of the blood out of the heart into the circulation, which is good. That's a healthy heart. And then the mouse with only 1 mutant copy, it can still do that. So it can still pump very well. It can still get blood to all of the mouse. That's good. But now, when we look at the mouse with 2 mutant copies, we see that it can no longer do that. It can still pump a little, but not as well. A lot of the blood just gets trapped in the heart, stays there, and so it's not going into the body. And so that's another clear sign that these hearts are not as functional, and so that they are not as good. And indeed, when we look at these trained mouse hearts, we see that the mice with the 2 mutant versions, they are even worse. Their left atrium is even more engorged, and their right ventricle, if we look at it a little bit closer, you can see on the bottom that these are these thick red streaks, clear fibrosis, a clear sign that this mouse is sick, and that this heart is kind of looking like what you would see in a human patient. All right, so that's already pretty interesting. But of course, the problem is a mouse is not a human. So can we maybe also create another type of model that is closer to the humans? And that's what we did when we tried to create human stem cell models. What is that? Well, imagine you have a real patient that has the disease with a mutant version. And we take some of their cells, and then we transform those cells in a Petri dish to tiny little beating heart muscle cells with that mutation. We then take some more of those cells, and we remove that mutation, we fix it. So now that these cells, again, more resemble what you would see in a healthy human, and then we also turn those into little beating heart muscle cells. And now we can compare these two and see what is the effect in a human context, what is the effect of having this mutation. And what do we see? Well, if we now look at the gene expression, we see, of course, that the fixed version has a certain expression of certain messenger RNAs. But now, the version that we took from the patient with the mutation, it's very different again. And this time, it's really, really different. There are a lot of differences in the messenger RNAs that it creates. So it's clearly the case that for the human cells, having 1 copy of this mutant has a much stronger effect than it does in the mouse. Okay, so if we now take this all together, we compare all of our models. What do we see? In mice with only 1 copy, we see a few genes that are otherwise expressed, but not too many. In our mice with the 2 mutant copies, we see that they have a clear, sick heart already from the outside, as well as a few more genes that are differentially expressed, and that after training, their heart function goes down quite a bit. And for the cell models, we see that there are a lot of genes that have this differential expression. Okay, so that's really interesting. But like I already mentioned, there's not just 1 gene that can give this disease, there's a whole list of genes. So we figured, okay, let's also make another model, this time in a gene called plakophilin-2, or PKP2. And this protein does not go across the membrane like the other one, but this is inside the membrane and it makes other connections to other parts inside the cell. So it's still an important part of that big, complicated, intercalated disc. So now we created the same types of models for this one. And again, we want to compare how do these models look when we compare them to each other. So when we have a mouse with one mutant copy of this PKP2, what do we see? Well, we again see that there's only a few genes that are differentially expressed. But we did notice that after we train these mice, their heart function also goes down a little bit. So it seems that when these mice are at rest, their heart is pretty happy, pretty healthy. But when we put in that extra stress of that training, like in humans, it really does extra damage, and now their heart is less functional. So that's also very interesting to see. But then when we looked at the mice with 2 mutant copies for this particular gene, we find that they all died. But they didn't even die as mice, they died as embryos. They died before they had a chance to be born. Because as it turns out, at least in mice for sure, you need a healthy copy of this PKP2 in order to just survive till gestation and to survive until birth. So we couldn't study these further, of course. But then we looked at the IPS model as well, that we created from another patient. And here again, we find that there are more genes with a differential expression than in our mouse model with only 1 copy. So again, here it turns out that in the human context, there are more genes that respond to having a copy of this mutant gene. Okay, so now we have these models, we can use these to learn about the disease, but of course, before you can fix a disease, you need to find it. But also there, ACM has to be very difficult and the diagnosis is very complicated. You have 6 possible categories based on imaging and histological analysis and depolarization and repolarization issues and arrhythmias and genetic history. And for each of these, you have major and minor criteria, and it's very complicated. So it would be really useful if we can find a way to make this diagnosis easier and clearer. But what could we look at? Well, we thought, what about microRNAs? Very short, what are microRNAs? Well, remember that your DNA can create all those little messengers to create proteins to go do stuff. Well, it can also create another type of RNA called microRNA. And this microRNA isn't going to go off and do stuff. Instead, it's going to regulate what the messenger RNAs can create. So it's not producing something itself directly, it has a regulatory function. Okay, but why do we care? Well, here's the thing. If we now look at the bloodstream of a normal human, and we see that in this bloodstream there are all these tiny bubbles, tiny vesicles, where all kinds of microRNAs are circulating. Okay? But now when we look at an ACM patient's blood, we see that they still have these little vesicles with microRNAs, but as you can see, it seems that certain kinds of them are much more present than in a healthy human. And indeed, when we then look in general, it seems that there's a certain microRNA called MIR185-5P that in general, on average, is much more present in an ACM patient than in a healthy human. And so now if we can test this in a clinic setting, if we can bring this to the hospital, and we can make an easy test out of this, that would be really good, because that would mean we can have an easier diagnosis with an earlier intervention, and especially for ACM, this is important, because then we can very soon start with those lifestyle changes. Because like I said, remember how normally exercise is good for your heart? In these patients, it's important that you find the disease early so they can at least don't do that kind of exercise, so they can prevent from accidentally damaging their own heart. And so it's really important that we find a good biomarker for that. All right, so now, in conclusion, what did we learn today? Well, we know that mice with 1 copy of the mutant gene, no clear signs of ACM on the whole, they're pretty happy mice, but it does seem that they are more vulnerable to conditions of extra stress. Then, mice with 2 copies of the mutant gene, it depends. If it's DSG2, they do tend to show ACM-like hearts, but if it's PKP2, they die as embryos. It's not compatible with life. Then, if we look at our patient-derived stem cells that have 1 copy of the mutant gene, we find that there are multiple genes that show a dysregulated expression. And then finally, we know that there is more, on average, more MIR185-5P circulating in the blood of these ACM patients, which means that it is a possible biomarker for diagnosis of ACM in the future. All right. Well, then I would like to thank you all very much for your attention, and I would like to give the word back to the prorector.
Thank you very much for this presentation. We will now go to the opposition, and the opposition will be opened by Professor Dr. Paula da Costa Martins, Professor Paula da Costa Martens is professor of molecular microvascular biology at this university, and she is chair of the assessment committee. Professor.
Thank you, Mr. Prorector. Well, dear candidate, first of all, congratulations for this very nice piece of work. And of course, your supervisors also for guiding you throughout the whole process. And referring to one of your propositions, I'm sure you knew all the time what you were doing, otherwise you wouldn't have ended up with this nice thesis. So I have a few questions I would like to discuss with you. Some points of the thesis, and the first one relates to the discrepancy between the genotypes and phenotypes that are observed in humans. So some people have the mutation, but they don't develop the disease or develop not such a severe phenotype. And also you try to mimic this in your models, so I was wondering, how are you going to really identify new mechanisms if the phenotypes are so different? There's so much variability. How are you going to say, this is the mechanism that is involved in this phenotype? Is it only one mechanism, even though you have only one mutation?
Dear highly esteemed opponent, first off, thank you for your kind words and for your question. So you're definitely right that it's in ACM very complicated, that you have this big discrepancy between what you see on the genotype level and on the phenotype level. And in order to study that, you can try to account for it. So for example, you can already try to have different genetic backgrounds, especially in an IPS context. You could try to say, okay, if we create 3 different IPS lines from healthy humans, for example, and from a healthy human we start and we introduce the mutation, and then we can already see, okay, is the effect of this mutation different in the different genetic backgrounds? Then I also think you can maybe go within certain families, and if you say, okay, these 3 people have the same mutation, one of them gets it really bad, one of them gets it a little, one of them doesn't get it, then you can maybe take a step back and see, okay, where else do they maybe differ somewhere in the genome, and then see, okay, this and this gene is a little bit different, a certain variant, they are connected to these pathways. And then can we maybe go back to our model and see, okay, are these pathways then also indeed activated in our model? And then hopefully also compare it to other existing models and try to determine, okay, if this is a pathway that is always disturbed in ACM, like a sort of common core, or is it possible that certain genes or maybe certain mutations for certain genes have certain peculiarities, so certain changes that are only really happening in that case?
But then you're saying that there are additional mutations in other genes.
Well, it could be genes, but it could also just be slight changes, just like your genetic background can differ. So even if it's not really like a pathogenic mutation, I think it's fair to say if you have 2 humans, one is healthy, one is sick, with in that one DSG2 gene, for example, the same mutation, yeah, there's a reason they're different. Could also be an environmental part. Could be that the one that gets sick was a real sportive person, and that's why it got sick. But assuming we can rule that out, it could also just be that because of some other variants in other genes that in normal circumstances aren't dangerous, that it makes them more vulnerable and more susceptible to the influence of that disease-causing gene.
Because then the same question would apply to the finding of new biomarkers. Because if there's so much variability, would one microRNA explain the whole thing?
That is also a very good question, because for the study we did, we started from a bunch of ACM patients that have like the classic ACM. And so it does seem that they all had different backgrounds, so it does seem that there is a certain amount, that in a lot of people that this microRNA is upregulated. I think it's likely that you won't find one marker that is always extremely upregulated compared to everyone. Also in our graph, you saw that there are still a few healthy humans that by chance have a little bit more of that 185.5p, and certain ACM patients that have it a bit low. But I think it's a bit, you don't need to find one marker that is perfect. Ideally you would, but if you can find one that can find 90% of your cases reliably, that's already massively useful in a clinical context. So saying, we're not going to look at it because we can't find a perfect one, I think you can try to find as good as possible, but if you find something that's usable at least, that has a good way to distinguish for most people, I think that's still useful.
Okay. Then I'm also going to go into your desmoglein homozygous knock-in mice. And the phenotype, because that's an example of a severe phenotype with this very enlarged atria. So I was wondering, so you chose to look at the mice at 3 months, and they already had a severe phenotype, and then you looked also at 6 months.
Yes.
I would think if I would see a mouse with such a heart, I would think of, okay, I'm not going to let these mice suffer even more. I'm going to stop the study. So what was your criteria to go on with the study and wait 3 more months? And also, why? I mean, the phenotype is very severe, and these mice still live. So what is behind it?
Well, it's actually indeed very striking that some of these mice, like you saw the trained mice with that absurdly engorged atrium, they were on the whole, like especially when the trained ones, I really kept close track of them. And in terms of like their behavior, they were really happy. So it seemed that, okay, we had a bit more drop-off with the homozygous knock-in, but on the whole, they tolerated pretty well. But one of the reasons that we also chose, to still go to 6 is because in the beginning, we figured we were only going to look at our heterozygous because that mimics the human disease, And you start looking at it and you look, okay, maybe it's not that much. But, in the meantime, you've been breeding more litters and you see, oh, okay, it looks like this homozygous knock-in is also viable. Let's also look at it because, why not? And then you start noticing, oh, okay, they already have something. But, because you also still want to be able to compare to your heterozygous, we decided, okay, we're going to keep the time points that we chose for now at 3 months old and 6 months old, so we can still compare all of our mice and that we don't, have a few at 3 months and then a few at 2 months, and then, it starts becoming very difficult to make good comparisons. And as it is, I think it definitely makes sense if this study would go on to also look in this homozygous knock-in mice at maybe an even earlier time point. And then depending on what you have access to, could be really interesting to see at exactly what time, for example, these atria start engorging. Because we also actually have a small side project where we have those embryos. And we also tried to, that we did for the PKP2, but also for DSG2, we tried to get some homozygous knock-ins. And the results aren't in yet, but it would be really interesting to see if already, like at gestation, there's some difference there, or if it's really that the mice are normal, and then only after they're born, maybe once the heart really starts pumping and functioning, that the damage gets in then. So that would be really interesting to see.
And how would you mimic then these phenotypes in vitro? Because you use iPS cells. Okay, it's nice because they're human cells and they already have the... But how would you recapitulate these phenotypes? Because, I mean, the phenotype is mostly atrial. You start with normal myocytes, and I don't think you differentiate them in... So how would you tackle that?
That is a bit of a problem currently, especially in our system. We still just did like 2D system with the iPS cells. You could go to a more 3D system already, but even then, indeed, you're still going to miss that complexity of lots of different cell types that you see in a real heart, and like the ventricle and the atrium. So that is indeed something that we miss. But I do think it's good to remember that while that big atrium is interesting, and we also see it in some other mouse models and even in some humans, I think it's fair to say that the disease is still mostly ventricular. So I think you can still use your iPS cells to study, the core part of really what drives the disease. But that is indeed where I think that we won't be able to get rid of the mouse models yet, because you won't right now be able to see things like that engorged atrium in just like a Petri dish, because we don't have systems yet where we have like a real beating full hearts in a Petri dish. We're not that far yet.
One last question.
Yes.
Just curious about the models, but since you found all those microRNAs, well, you found 185 as the main, but you also found other microRNAs that were differentially present. Did you try to validate that or try to see if those microRNAs were also differentially expressed in your mouse models and your iPS cells?
We didn't get that far yet, but it's definitely something I would also like to look at. But you always have to choose. You can never look at everything you would like to look. So we didn't really check much in regards of microRNAs in these models yet. But now that we have a good basis, that we know what is happening, like on a big level, now indeed it would be interesting to take a step and go even further. We have certain RNA-seq data that I think could also be used to get some microRNA information out of. But again, we got that pretty late, so now we just wanted to focus what are like the major changes, what is the big thing that's happening, and then we could indeed look at maybe what is exactly driving these changes, and then maybe certain microRNAs that are up or down, and then does this correlate to humans or other mouse models.
Well, thank you very much for your discussion, and I will give my word back to the prorector. Thank you.
Thank you. The opposition will be continued by Professor Dr. Paul Volders. Professor Volders is Professor of Genetic Cardiology at this university. He is member of the Assessment Committee and he is also Secretary to this meeting. Professor Volders.
Thank you very much, dear Mr. Candidate. This is a very nice thesis. I congratulate you. I think it's something I read with pleasure. It is of high scientific value, not just for mice, but also for men. And of course, I extend my congratulations to Professor De Winde and Dr. Calora and your family. They're part of the journey, I think. Right from the start, you discuss the evolving insight that arrhythmogenic cardiomyopathy is not necessarily confined to the right ventricle, because it may also be primarily confined to the left ventricle or maybe biventricular. Now, if you go back in history, and especially if you read your own sentence on page 9 on the triangle of dysplasia that was introduced in 1982 by Professor Frank Marcus and colleagues, then you write the following. The triangle of dysplasia is between the inflow tract, the outflow tract, and the posterolateral LV.
Yeah, that's should be RV, I guess.
Oh, that should be RV.
Yeah,
okay.
that's a typo I actually noticed after the print was already out.
Okay.
That's a mistake.
Then I'm happy we corrected that right from the beginning, because otherwise I'm not sure Professor Marcus would be happy with your citation. Yeah. Okay. For the purpose of this opposition, I went back in my own registries, because I'm a clinician. I see patients with arrhythmogenic cardiomyopathy, and they are very variable in their phenotype. In a way, this relates to the discussion with Professor Da Costa Martins. Roughly 300 patients that I follow up, and when you classify them exactly in the way that you did it on the last slide or so of your presentation, the 6 clinical criteria, here in Maastricht, probably we have 10 cases where biopsies were taken out of the 300. Other centers do this much more consistently. They go for a biopsy with everyone. Now, if you and I would walk to the Department of Pathology to convince them to get these biopsies from the hearts and to do staining or any characterization at the tissue level, what would be your most convincing argument on the basis of this thesis? What would be the best reason to explain why tissue characterization in connection to the other clinical characterization is super relevant to execute, even if it's dangerous in terms of complications?
Okay. Dear highly esteemed opponents, thank you for your question. I think one of the problems with ACM diagnosis is that it's also that there are other cardiac diseases that have a lot of overlap with it. And I think that's why taking that biopsy is a bit like, if you really want to make sure that it's ACM and not something that's related, like another arrhythmogenic problem, that's why you need to take that biopsy. But then, of course, like you say, there's possible complications. It's not as easy. So, I think it's right not to do it unless you really need to be sure. So it's relevant to do it, but if you can avoid it, if you can find an easier way to exclude other cardiac diseases and say it's definitely ACM, I think that would be really good.
But that answer surprises me, because in all of your data that you show, where there is so much histology, so much immunohistochemistry, so much molecular data, everything you could do on those biopsies, you're now saying, well, save that for the last step in the characterization in the clinic. Whereas in the clinic, the phenotype is variable, difficult, has a long course of development, may or may not be arrhythmogenic at some point, may or may not turn to heart failure, may remain without problems, and then people die at a normal age. So in that sense, I find that a little bit contradictory.
I understand, but then if I may put it a bit crudely, I'd rather, do histology on a whole bunch of mice and then see, okay, how did their heart look on histological level? And how can I link that to how their ECG looked, how their echo looked, and then learn about that so that I don't have to then do it in a human patient. So that's why we study it in the mice in the first place, so we can learn what we can look at and what we can link, how we can look at certain parameters, and then confirm that it's the disease without having to do much histology in human. So I don't think that's contradictory, because in the end, a clinical setting, It has different priorities from a research setting.
Okay, this interesting discussion gets more interesting with the minute. Let us discuss then what is the life course of these patients. So they may turn 80, no problems. They may die suddenly, roughly at the age of 20. And then there is the gray middle portion that reaches an age somewhere 50, 60, and may die of heart failure. So it's a big spectrum. But essentially, the problems in patients usually start at adolescence at the earliest. So the first question there is, why only at adolescence? Because there is, let's say, 10, 15 years of delay in a beating heart with children playing, exercising, etc., where you have explained to us already that the genotype is there from the earliest form of life in that same individual. So what is explaining the delay of the phenotype?
Well, I think that right now in ACM, that is one of the big questions we're still trying to solve. Why is it that there's this delay?
But you have the mouse model to answer it.
But yeah, so we can use this mouse model to see why it would be. But right now, we don't know for sure yet what is driving it. We know that the training, for example, made it worse, and we know that in humans that makes it worse. We also know that in humans, there are some cases where you don't notice it, until the person drops dead.
Okay, and that is rather drastic also for the family. So considering then this pretty sudden demise of those who die suddenly, then you can make a distinction between an arrhythmogenic form of the cardiomyopathy, where there indeed is an early sudden death because of ventricular arrhythmias, versus those that survive the phase of arrhythmogenic vulnerability and go into the failing phase, if at all. In the studies that you showed in the mice, it's mainly the structural cardiomyopathy that you describe, I think, but I may be wrong. You may correct me there. Do you feel that the mouse models that you showed in this thesis do have sufficient representation of the, let's say, arrhythmogenic version of the disease?
The thing is, we did some ECGs, but only at later time stages. I think it would be interesting if you could take, especially these homozygous knock-in mice, and at a very early age, as soon as you can, they have these little implantable ECG monitors, and then we could see, indeed, before this mouse starts showing overt signs of ACM in the heart with the scarring, do they also start getting those arrhythmias or other problems? And that would indeed be interesting, because now that we know that they do it, and now that we know that at least at 3 months old, there's already starting to be a problem, it would be really interesting to see. The thing is, the ECGs that I did, they were already at a later stage, and they were also not the greatest system, to be honest. But it would be interesting to indeed see, do the mice only recapitulate this later stage of the human disease with the overt damage in the heart, or do they also already have problems with conduction before these hearts start really showing problems?
I think that is a very important answer, and you especially please the arrhythmologist in me, so that is something very important. I look to the prorector. Do I have a short second for a final question?
A short question and a short answer, please.
The short question is, how relevant is a homozygous model of a mouse to the heterozygous human situation?
Well, we know that there are some cases for ACM that there are also patients with homozygous mutations, maybe for other genes. So it's not perfect, but I still think it's relevant to teach us something about the disease.
Thank you.
Thank you. The opposition will now be continued online, and the opposition will be given by Professor Dr. Brenda Geroul, and Professor Dr. Geroul is Professor of Cardiovascular Genetics at the University of Würzburg in Germany. And I have to add, member of the assessment committee.
Yes. I also read your thesis with much interest, in particular about the different mouse models you have generated and also characterized. And so I will focus a little bit on the maybe plakophilin 2 mouse model, because plakophilin 2 is also my great interest in research. And so I found that model very interesting, but could you maybe explain or maybe similar to previously published plakophilin-2 mouse models, and maybe what are the advantages of your mouse model, maybe compared to other ones? So that's maybe my first question.
You're right that there have been a number of other mouse models for PKP2. I don't gonna say that I know all of them by heart, but I think ours is very interesting. We definitely know that it's from the human context that has that disease with just the one version of the mutant allele. I think there are some similarities in terms of the problems with the channels and the conduction, because from what I remember in other models for PKP2, something that they often saw was like problems with connexin 43 expression or localization, and then other ion channels. And I think that's a part where their models more or less overlap, not always in every part, but I think when it comes to the PKP2 models, that's an angle that's really interesting to study with this model.
True. So maybe you can in general say what kind of mechanism or genetic mechanism is behind plakophilin-2 mutations. So yeah, so maybe, so is it, do you know what I mean? So if you look in humans, what kind of mutations are mainly causing the disease?
If I recall correctly, in humans, for PKP2, right? That there's a lot of, like the one we have is like nonsense mutations.
Exactly.
I think so.
Yeah. And what is the consequence of nonsense mutations?
You get a short part of the protein. And so then, you know, you have only a little bit. Then it can also be, because you have a nonsense mutation, that you can have nonsense mediated decay, which is then a bit depends on your context, because that's something we saw in our mouse model. We think, we didn't really confirm, but that does seem to be, for example, a big difference between the PKP2 mouse model and the cell model, where in the mouse, it seems that the mutant PKP2 mRNA is really degraded and suppressed, while then in our human context, it seems that it's much more expressed, so it doesn't seem to be as degraded. And I think this could be relevant for also the human disease, whether then, you know, you just have a mutant that gets degraded, and then you might have that the wild type that's enough is not enough to perform all the function, versus another case where the mutant is present and it really has like a dominant negative effect by pairing with certain, partners, but then not doing the function it's supposed to because it's missing the rest of the protein. And so then the native healthy PKP2 cannot perform its function anymore.
Exactly. So here we have a loss of function mechanism in PKP2. So that was my answer I wanted to, but you explained it right. So so going to the arrhythmias and going to the connexin, maybe a more special question. So what you have seen is that connexin 43 is downregulated. And this has been seen in other PKP2 models before, and you already mentioned that. Could you maybe explain a little bit broader what that means and how this is maybe leading to arrhythmias?
Well, of course, connexin 43, it's those little gap junctions, and it's important to make sure that conduction passes from one cell to another as well. So I think if you have less of those channels present, that you will just not have as good of a conductance throughout your heart as you should have, because they are lacking these connections.
Yes, but what is the role of PKP2 in it? So why is connexin 43 maybe downregulated?
For one, PKP2 is one of those part of the intercalated disc, and a lot of these elements of the intercalated disc are connected together. So if there's a problem in one of them, like they don't dissociate, they can really, the whole structure can kind of fall apart. So it can have far-reaching consequences to the expression or the localization of other elements. So even if you still have, I think there was a model where they saw, I think it's a PKP2 model, where they saw that connexin 43 was still equally expressed, but it just didn't go to the membranes anymore. They had mislocalization. So I think that's a big part where even if it doesn't mess with the amount of the other parts that are there, if they cannot connect together anymore in this big complex, they don't get where they need to be, and so then they might as well not be there.
Yeah, that's a good thing. And then one question maybe related to your mouse model. I think that was mentioned before. So you just did an ECG on your mouse model, but how could you maybe accelerate arrhythmias if they are there? So are there any experimental approaches maybe to get them more prominent?
Can use to really induce those arrhythmias, if I recall correctly, so like isoproterenol, I believe you can do, and that can really push it. And then indeed in certain contexts, they've done that, where they see, for example, the mouse has okay conductance, but when they add that extra challenge, then the wild type mice respond better, and then the mice with the mutant PKP2, they can't handle that. And so again, here we see that at rest, at homeostasis, it looks okay, but then when there's this extra challenge, this extra stress on the system, then the mutant animals or cells cannot handle that stress anymore.
Yeah, that's one possibility. Yeah, you can perform. Okay, so then I was a little bit. So you told in your thesis that you also investigated age of mice, and then I was wondering, because you termed the age 6 months. So is this really a high age of a mouse, or maybe is there another option?
Yeah, so 6 months old is not that old for a mouse. I believe there were other mouse models without training where they had to wait until 9 or 12 months old before they really saw it, because we know it is a progressive disease. So you could say that 6 months old is maybe a bit too early. But here we're also, for the training, we had certain limitations because I had to go on a secondment to do this training. And, I only had so much time. And I literally finished the training of the last mice and then the echo and the ECG like a week before I left again. So I got the maximum age that I could practically manage. But you're right that maybe even higher ages might be might teach us a bit more or something else. But yeah, we had to work with what we could.
Exactly. So in an ideal world, we would wait longer and then just do some of the examinations again at a later stage, in particular in those heterozygous mice. Okay, I'm done, or because
if you have a short question, you may ask it, and if not, we can conclude. Okay?
No, it's fine. I can ask one other short question. So now you have a mouse model where you have the same background, which is a good model to study, because you said in the patients they have different backgrounds and that may influencing the phenotype and so on. So do you have any other idea what kind of triggers you can apply in addition maybe to endurance or exercise to accelerate the phenotype?
So while staying with the same mice, you mean, or changing in the background? Because even if you don't want to create a new model, I do know that there have been studies where they have a certain mouse model with a mutation, and then they backcross it into another background, and then see after a while, does this new background make a change? Because there was one mouse model, was for another gene, I believe, where they had one allele was completely knocked out. In their background, they saw that it had quite a high, that these heterozygous had quite a high die-off. And then when they started backcrossing into a new background, they saw that this lethality dropped off very quick. It dropped off quite a bit. So there they had a fairly cheap and easy way to see what happens, what is the effect of this mutation in another genetic background without having to create 3 more mice models.
Exactly. I guess that's a huge reason why there are different mouse models published with different phenotypes, I guess. So you see the influence of mouse background on the phenotype already. Mm-hmm. Thank you.
Thank you, Professor Grol. The opposition will be continued online by Dr. Paola Braghetta. She is Associate Professor at the Department of Molecular Medicine of the University of Padova in Italy. please.
Hello, good morning to everyone, and congratulations for the very nice thesis. I did appreciate it very much and enjoyed it very much. So I have a few questions for you about mainly the first part, the desmoglein 2 mice. And you mentioned the histological differences you found in the mice, in the heterozygous and homozygous mice, but these do not recapitulate the phenotype you have in patients. For example, you don't have any adipose tissues, and you mentioned also some calcification, but I didn't see any histological analysis confirming this. Can you comment on this, please?
Dear highly esteemed opponent, thank you for your question. You're right. To be honest, we did the staining for with the fibrosis, and we didn't really get to the point to really make sure exactly is it calcification or fibrosis or what exactly is going on. But we do see that there's a clear scar. And then for that lipogenic, that's a known problem with mouse models for ACM, that it's very difficult to have this fatty part of the fibrofatty replacement, because in general it seems that mice hearts, because they're not human, are not as prone to getting these fatty deposits. So that's something that indeed we don't see in our model. But to be fair, that's something you don't find recapitulated in a lot of ACM models.
I see. And what is your explanation about this? I mean, what's the differences you think that are between human hearts and mouse heart? I mean, for example, what sort of cells could be activated in one situation and the other one are not activated, or something like this?
Well, from what I know, if I remember correctly, and it's also not entirely clear yet in human hearts what is actually giving this adipose tissue. So there's been some thoughts, some papers thought that it would be cardiomyocytes dedifferentiating, but that seems rather unlikely. It seems to be other cell types in the heart that are either already in the heart or possibly even certain cells that migrate into the heart and then create this adipose profile characteristics. But in the mouse, it doesn't happen. Yeah, you can just point to the fact, a mouse is not a human. It's a bit vague, but then I think in terms of specific pathways, I'm not 100% sure, because I do know that a lot of the pathways that are relevant in humans, like the Wnt pathway, we see the same thing in a mouse. But so then why doesn't it also create that adipogenic? Then, yeah, it's just not as prone to it. I'm not 100% sure the exact molecular mechanism behind it, because I do believe there's one or two mouse models where they find some lipogenic presence, so it's not completely impossible, but it's just not as prone to creating that in general.
Yeah, and you mentioned that the pathways that are activated in these pathologies. And what are the most relevant ones you think that are involved in the onset of the ACM? And or at least what would you focus on in your future studies, if you have some special ideas about it?
So in terms of the major pathways, I believe that the Wnt pathway is usually downregulated, while things like Hippo are upregulated. Then I believe that TGF-beta is usually also upregulated in ACM, and then something GSK3 beta, I believe, because I believe there are also other models where they studied that and where they found that a GSK3 inhibitor could actually rescue a lot of the ACM-like phenotype. So I think that's already an important pathway to look at. But in general, there are multiple pathways that you can look at, and the problem is a bit for example, most mouse models, if they have a change in the Wnt pathway, it goes down, like in human. But then there are some mouse models where they also have an ACM-like phenotype, and they found that the Wnt pathway is upregulated. So it's already not as easy to say, yeah, if you see that it's down, it's good. It's not all that obvious.
Yes, this is absolutely true. And whenever you activate a pathways, you know that the genes that are modulated are quite a lot. And some of the genes are really very sensitive to these pathways, as you said, for example, TGF-beta or YAPTAZ pathways are the extracellular matrix genes. Do you know something about the extracellular matrix in your heart, in the mouse of your models?
Excuse me, could you repeat the last part of the question? I didn't quite get it.
Yeah, if you know something about dysregulation or modification of the extracellular matrix in the hearts of your Gnocchi mice.
In terms of that, I think most of what we have as evidence is the RNA-seq data. And from that, we did find that there are a lot of terms where it's like, changes in the extracellular matrix in some of our models, but then we also saw that it's not recapitulated in all of our models, but we haven't looked at it in great detail yet.
So I think this is an important point, because the extracellular matrix is really changing the behavior of the cells. So, I mean, it's just to have a look at that is really quite important. Another point is you mentioned many times in your thesis inflammation, and you show some genes that are dysregulated in your models. But what do you think is the importance of inflammation in this pathology?
So here in ACM, you see that in a lot of human patients, there are these inflammatory infiltrates, but also here the exact relationship between inflammation and ACM is not 100% clear. As far as I know, what it seems to be now is that the heart starts with that it starts getting damaged. The cells create certain signals that then attract these inflammatory responses. And then this inflammatory response can either maybe even do more damage, because then there's like a crosstalk between the cells that arrive there and the existing cardiomyocytes. But it's not 100% clear, because it's also the case, if I'm not mistaken, that in a lot of ACM patients, you see that there's like this infection with a virus in the heart. And then one of the questions is, maybe it's not the case that the inflammation responds to the damage. It's more that the damage happens. It makes the heart more vulnerable to these infections. And then these infections are what call up the inflammatory response. And then that inflammatory response can still do, further damage the heart. But so I think it's clear that the inflammatory response is relevant for the disease. We don't really know yet exactly at what point does it come in, does it always come in, and what is the exact role that it plays.
Okay. And would you think it would be interesting to investigate this inflammatory part of the disease, and how would you approach this?
I definitely think it would be interesting, and this is one of the cases where I think the mouse models are, you better to study it than the cell models, because in the cell models you can maybe find, okay, the cardiomyocytes create this interleukin that can attract certain immune response, but then the actual immune response, you have to see it in a more complex model. So we saw in some of, not too many, but in some of our histological slides, we saw this infiltrate that reminded us that we think is an inflammatory response, but also here, these are samples we got fairly to the end of the project process, so we couldn't look into it more yet. But it would definitely be interesting to at least start looking, okay, exactly what kind of inflammatory cells are here, then maybe also do, like on a qPCR level, see, okay, which interleukins are more expressed, or which might they be attracting, or how might they be talking to each other.
Okay. And I mean, if you would think about a therapeutical approach to, not to cure, but at least to slow down the progression of the disease, would you think how can you think about a molecule or a group of molecules that could be used in this thing?
You mean specifically with regards to the inflammatory angle, or in general?
In general.
Please, a short answer.
Well, like I said, certain pathway inhibitors, like the GSK3 inhibitor, I think there's a lot to be gained there. But then the question is also how to make sure that it's specific in the heart and that you don't, have a systemic side effects. But if you can do that, that I think has a lot of potential.
Thank you very much.
Thank you, Dr. Braghetta. I forgot to mention that Dr. Braghetta is also a member of the assessment committee. The opposition will be continued online by Professor Dr. Jolanda van Hengel. And she is professor and chair of the Department of Human Structure and Repair at the University of Ghent in Belgium.
Thank you, Chair. Thank you for adding me to your committee, to your defense. And I read your thesis with a huge interest. Okay, so I have still some questions. A lot of questions already done, but still. So I will start with your introduction. And so you are talking about intercalated discs. You are talking about desmosomes, adherence junctions. There are also hybrid junctions. So you showed us a scheme. Can you explain better how the intercalated discs are made and what you can find back in the intercalated discs?
Dear highly esteemed opponent, thank you for your question. If I understand correctly, so the intercalated disc is basically made up out of multiple components, which also can be part of other things. So desmosomes occur in the intercalated disc, but you can also have desmosomes in other contexts, so it's not exclusively there. So you have the desmosomes, there's adherence junctions, and then those gap junctions. And then indeed in the intercalated disc, which is like kind of specific for the heart, they really all come together in one big complex, and there's a lot of crosstalk there.
And so this is then the hybrid junction. Do you know who invented that name and how they were seen?
No, I have to admit that is not something that's readily available. I'm not sure the name of the person who found it.
And the technique, how did they find it?
I remember that one of the, I'm not sure if it's how they found it, but I remember one of the things is that they found that like on I think electron microscopy images, that they have these very dense clusters, and that you can really see over the heart that, you know, there's really these nodules where all these things come together. And that made it clear that these intercalated discs, you know, it's not that the entire wall of a heart muscle cell is connected to the entire wall. It's really like these small nodes. And indeed, we also did a bit of electron microscopy in the project. And indeed, you see that you have like these two membranes next to each other. And then you have this very dense area, and that's where those intercalated discs are.
Yeah, okay, indeed. So then about desmoglin 2 and plakophilin 2. So they are not exclusively expressed in the heart. So you mentioned and you looked to the heart in your mouse models, but what do you see more in your mouse models? Or is there something more to analyze, maybe in the skin or in another organ?
Indeed, especially in the beginning when ACM was discovered, it was really linked with certain skin issues. I think the Naxos disease is a classical one, because indeed also a lot of those proteins come back into those skin cells, which also need to make these tight connections. I checked the skin in the sense that, you know, when I was harvesting, I always checked if I could really see, is there already a clear problem with the skin? I never found anything. I did often, you know, get a piece of skin and fixated it anyway, just in case we found something later, we still wanted to look at it. But at least, you know, outwardly, I never saw any mice which had like problems with their skin or their fur. So it seems that in mice, there's no real clear connection. But to be fair, there's also a lot of ACM patients that don't have problems with their skin. So it's really just a few genes or mutations that give that.
Yeah. Okay. Then, so we were talking already, or you were talking with some other opponents about dominant negative effects and shorter proteins. How sure are you? Are they present, and how will you analyze that? What do you need to be sure that a smaller protein is present or not?
So that's actually something we really wanted to do. We tried a lot because it would be super relevant and interesting to see, okay, the mRNA does this, but what does the protein do? We actually had a company create a custom antibody for us that should only be able to find the shortened peptide, but we didn't get it in time because they forgot to work on our order. So that was very unfortunate, but we could definitely look at that. We could also maybe do some like mass spec to see, do you find like these peptides only, where there's only this little parts present. And then if you can confirm that this short protein part is there, I think it would be interesting if you can also do some sort of pull down where you can see, does it still interact with certain partners? Because then you might see, okay, this might be why this small part of the peptide is creating problems, aside from haploinsufficiency issues.
Okay, so this is still on your program, I suppose. So you did also some work with the induced pluripotent stem cell derived models. Can you okay, you're talking about cardiomyocytes. Do you know how we can make like atrial cardiomyocytes? Do you know the difference between the protocol you are using and the protocol that is published?
I'll admit, I was, in the IPS story, I was more on downstream, so I didn't really do much for the creation of them. So I know the protocol they did is then have like cardio selection medium and Wnt signaling, but I'm not really sure about the details about how to make it more atrial versus ventricular, I must admit.
So for the mouse models, you try or you use these endurance models. Can you do a similar thing for the iPSC-derived models? Did you think about it, or just think about it, discuss about it?
I know that there are, well, maybe in 2D it's a bit less easy, but you can have these spacing where you force the cells to beat at a certain angle, and if you at a certain pace, and if you can get into 3D models, you can really have these small parts that are really beating active.
Robin Kolpaert, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. And I request you and your company to await the results of our deliberations and our return to this room. Thank you.
Please all rise.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
My goals only getting clearer, east side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. I know what's on the line. I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer, east side to the west side. No place like home. If there's questions that you've got, go the extra mile.
Because I wasn't looking at you, I saw you like looking up.
Long road, I don't waste no time. Break rules because fake this side. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad brands break it off. No room for negativity, praise and love. Prepare for deep part because we're taking off. And even though these nights seem long, I know that I'm never on my own. Know you won the battle once you grow.
How do I make the IPS more atrial? I need to know. Yeah? I don't know.
There are some.
I know, I'm sure it can be done, but it's like.
All the things you
Long road to the south side, 10 miles in my rearview mirror. I
Not a lot
know
of like discussions about
my goals only getting clearer. East side to the west side, no place like home.
I noticed that, but I'm like,
Questions that you've got.
I'm too short.
Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side, no place like home. Questions that you've got. Only takes a mile to die. Years can't fly by, don't waste all your time. Cause I'll go the extra mile.
Okay, two of you also, just one.
Yeah. You never got one. Just don't have the title in there.
Yeah, all the poses were practiced.
You got the phrase where people were in line after.
Yeah, it's very nice.
Yeah, it seems like I am for the pose. Okay, you should listen me and then I was giving a lecture. Yeah,
I can do that. It's nice. But the color is so bad that every picture I'm taking, either I'm adjusting for this or his face, this space, and it's all gone. Are you finished breathing now? Feeling better now? You know, I was in the office. I printed it there.
No, but the picture and everything. I went to Emma, so every picture is the same. I already moved everything. So
I
yeah,
will sign it.
yeah, you have your picture already. So you just have to.
Look like kind of jackass in
Nah,
the picture,
no.
but hey, for here.
I also have a lot of stickers for you to personalize.
It will annoy Robinson a little bit.
Mine has a lot.
You know those Instagram reels where the dog is completely covered?
We also found some below this photos that we saw on camera.
I have a picture with that. I forgot to send it to you all. Remember we went for that museum night? There's one with Robin with all the high collars.
But I have the one that we took from the... saw it the first time we were like in the South.
Yeah, but that was the one I meant. Like in the selfie machine with Hannah and
Yeah.
Eva. Oh, you have it.
Then you can...
I probably sent it like that day, but
I think
I didn't put it into the group.
there are too many things.
Did you skip it all?
That was still working on it.
But such a shame, both of you didn't get called. All kind of started on it, and I thought, okay, maybe.
Nah.
Grace didn't get called now.
But I didn't mind.
Because I was like, Paul was talking, I was like, oh, maybe she's going to go for it.
But after full body, I was like, the other ones want us. The other ones don't know that they can ask. The other ones haven't even looked at the positions. So I was like,
I'm done for the day.
but I was kind of like told that myself. Not of spite, but you know, like just to like.
So my question is, question and I was like, okay, now I'm proud of myself. I think that was the only question that they had from the 6th chapter. All his questions was literally the same we asked during the
I mean, to be fair, I think there were quite a few kind of
either rephrased from the mock defense
or elaborated a bit, but there were quite some of them. But I think to be fair, a lot of how they join right? Because it's like, first of all, want to hear what the other one has of questions. So there's a bigger list. Right now? No, not right now. Right now, they're doing before.
Yeah. So didn't you know? So after 1 hour, they come to say like the night is over and you have to.
That I knew.
What is her function?
She's the one who's like guiding everything.
It's not the professor.
No, she's not a professor. She's hired by the university and she's
Working.
yeah. But
And
she's
you have work a lot of, eh?
Yeah. We were just like sitting or so we thought at first that they would ask us to read some of these propositions.
you didn't have to read. You also didn't want to read.
I have mine in 1 month and I hope I do as good as you.
One got called up to me, but I don't know if it got read.
You the one but then you read it in English.
Maybe it was Robin that I know. I'm not sure. Actually, there was one, but
No, with Robin.
I think you had to read one from J.K. Rowling.
yeah, exactly. Because they were like, there's a lot of Harry Potter references. This was actually the Mystic
I really like your. Thank you. Refresh for this summer.
Yes.
But there was a slip. I mean, they won't see, they just hear.
Thanks to the tech support.
Get back, get back.
Robin Kolpaert. The degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor De Wind is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Please, all rise.
Dear candidate, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible? How do you answer?
Yes, I do.
Nice. I'm very happy that you said that, because then, by the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Robin Morris Wim Kolpaert, the degree of doctor and grant you all rights attached by custom and law. As an evidence of this, I now present you, in 2 seconds, with a degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Dear Dr. Robin Colpaert, it was a true pleasure to have you as a PhD candidate, and I was truly impressed with the performance today and truly impressed with all the efforts that you did in the past years. But I think for the last words for today, it's more appropriate that your direct supervisor gives you the classical laudatio. That's Dr. Martina Calore.
Dear Robin, I'm really glad that we are here together to celebrate one of your biggest achievements, and I'm proud for having been involved in your adventure. We met for the first time already more than 5 years ago when we invited you in our lab for the interview for the PhD position. There were 2 more candidates, and all of us were impressed by this skinny, nervous, and well-prepared Belgian-Flemish guy. You gave a beautiful presentation on your thesis and your interests, which included not only molecular biology, but also bioinformatics. And you addressed very well the questions from us. The choice was easy for us. You were the keen guy, good at presenting and at discussing scientific aspects that we were looking for. However, you know that bureaucracy slows down everything, also in the Netherlands, and we had to wait some months before your contract was ready. I remember I received at least 3 emails from you to get the contract signed. And with this, I started to have the idea that you were also a very organized person, so you scored another point. Soon after, the contract was signed, and you were eager to learn everything new. I still remember you following me everywhere with your clipboard, taking notes in a comfortable way with your cryptic handwriting. That you take things seriously became quickly clear, not only for working 100 hours a day, but also for arriving soaked wet in the lab when you chose to cycle for 25 minutes under the heavy rain, no matter what. Besides working in the field of molecular genetics, you were and are also into bioinformatics and programming. The stereotypical computer scientist can perhaps be described as an antisocial geek, but you're definitely not one of them. Your social engagement with people inside and outside the group was remarkable. You recruited people for lunch every day, and were an active player during the fun moments outside the lab, including even being a terrible karaoke singer. All in all, you have a habit of going out of your way to help others, and I admire you for that. That is exceptional and tells a lot about your personality. During your PhD project, you were working on characterizing different animal models for arrhythmogenic cardiomyopathy. Soon it became clear how hard this task was. Not only you had to learn novel skills, but also your life was made harder by the fact that we didn't get any of the results we were hoping for. So that we started to think of calling the exorcist. However, from a quick question to another small question and the gradual invasion of my desk, you started to get interesting results and being noticed. Indeed, you got sticked in the Drilichten grant and flew to Vancouver to induce physical stress to the animals. I know that you had a great time there, you did a lot of sightseeing, and you easily made new friends. I had no doubt on this. But I know that also you spent a lot of time struggling with the ECG tower and the echo that you performed on these animals. And still, most of the mice didn't show relevant signs of the disease that we were looking for. We will never know if, in the end, this is really true, thanks to our dearest FedEx that lost and ruined all your precious samples.
Yeah.
However, no matter what, Robin, this is beautiful work. You gave outstanding contribution to the complex field of arrhythmogenic cardiomyopathy and highlighted the readouts that could be used to test novel therapeutic approaches for the disease, which will be the next step in the story to which you also now belong. Congratulations, dear Robin, with this splendid achievement. Or I should say, proficiat Robin met jouw prachtige prestatie. It is my pleasure to extend my congratulations to your family, Paranemes, your colleagues, and to your many friends here. I'm sure they are all very proud of you, as I am. It has been a great pleasure and an honor to be your supervisor, and I wish you all every success with your life and with your career.
Thank you. Dear Dr. Kolpaert, also on behalf of the Maastricht University, I would like to congratulate you with your distinction, your degree, and we enjoyed the quality of your thesis and the enthusiasm with which you defended it. The committee was unanimous about that, and we wish you much of luck in your future career. I don't know whether you will stick to this hearty topic in the future, but for so far you made a great contribution. Thank you. And I would like to thank the members of the committee, especially those who joined us online. And we are going to thank the young doctor in two parts, one by the members online. And in the second part, there is a reception in the Refter, and you can follow the Beadle to the reception. And when you go to the reception, we will stay here to make a photograph with you, the members of the committee, the promotion team, and also the members online. And then there will be a photo on the stairs. So you will get lots of memories for this day. And with that, having said and nothing missed, I don't know, I close this ceremony.